Welcome to Biology Made Easy. Today we are starting our lessons on DNA. So in this lesson, objectives, we hope by the end of the lesson we would have known something about DNA functions, DNA structure, how DNA is seen in cells as chromatin or chromosomes, why DNA is universal, what makes DNA different in different organisms, which cells of organisms have DNA. Welcome. DNA is a genetic material, meaning is the material that determines all the characteristics of organisms. This is DNA, the structure. Well, this DNA determines structural, anatomical, morphological, what is inside, what is outside, the behavior. So this peacock is shaped, it's being a bird to start with, the shape, the colors, its behavior is DNA. This aquatic plant, this dog, this cat, their looks, every characteristic of this organism is because of DNA. And DNA, we've given ourselves the different shapes in which DNA can be presented. The DNA does not only determine the characteristics of these organisms, the DNA also is the structure that enables the organism to pass their characteristics on to their offspring. So this DNA enabled this bird to have these bird offsprings and not offsprings of another organism. And the behavior and everything is because DNA enabled the formation of the parent bird to get to form the offspring. The same thing, example, DNA enabled this cat to have baby cat that is a cat, not a mouse. It has all the cat features as well as behavior of a cat. And it's because of DNA, this molecule, DNA. Well, let's talk a bit about the structure of DNA, even though we'll look at the detail of it soon. So the DNA is a macromolecule. Macro means a very large molecule. But this large molecule is made up of 4 units. And each of the 4 units, we have 1 here, 1 here, 1 here, 1 here, 4. These 4 is made up of phosphates, sugar, and it's a ribose sugar. It's a 5-carbon sugar. And it has a nitrogenous base, a base of nitrogen compound. And the nitrogenous bases, there are 4 types. One is adenine. We'll look at the detail in a good time. Guanine. These are 2 rings. You have thymine and cytosine. Now, these nitrogenous bases are different, but the phosphates and the sugar are the same completely. And the sugar and the phosphate form a chain, and the chain has these nitrogenous bases taken at their sides. So you have the sugar and phosphate forms a chain, a continuous long chain with the nitrogenous bases taken at their sides. And you have one long chain, and there is another long chain, and this long chain is a complementary of this, meaning that the nucleotides have a way of pairing up. Adenine always pairs with thymine, guanine always with cytosine. And that is how the chain builds up to form this huge molecule. It's a polymer of nucleotides, meaning it's just the repeating units of these 4 nucleotides. It's a polymer. These 4 nucleotides, just the repeating. And there are changes. Adenine and thymine can come. The next block can be guanine and cytosine, or it can still be adenine and thymine, even in next three. So it's just repeating nucleotides that form a huge molecule of DNA. That is simply the structure. We'll discuss the structure in detail at another time. Now, the next thing to think about is, how is DNA seen in cells? Well, the DNA, when the cell is not dividing or preparing to divide, the cell at that stage, we call it interphase. At that time, we can see the DNA as chromatin material. See, as threads, just threads wound up in the nucleus of a cell, chromatin. Look at these cells here. You can see this is the nucleus. So the chromatin is DNA, which looks like threads with beads at various points. But when the cell is preparing to divide, this cell is preparing to divide, or when the cell is dividing, then the DNA is seen in the nucleus as chromosomes. See, so here is the same cell. Here is now a chromosome. This is a chromosome. This is also one chromosome. All right. Now, we will have to find out why it happens like that. Here you have a chromosome enlarged. This chromosome is one DNA, the same one DNA that you saw as chromatin. When the cell is dividing, you see it as chromosome. So let's explain what happens. First of all, chromatin. Chromatin is DNA in the nucleus gets support from histone proteins. So the DNA have proteins, the histone proteins form like scaffolding on which the DNA winds itself. Scaffolding is just like you have this bridge, and the DNA molecule wraps itself around the histone, which serves as the scaffoldings. And the whole structure, because it is very thin, is seen as a thread, like chromatin here. So that is chromatin. DNA with this histone, a thread, looks like a thread like this. Now, looking at it like this, you see, this is a nucleus of a cell, and this inside it is the chromatin. Now, when the chromatin is explored, here is a chromatin being opened up to see what is made up. You see, it's called around, this is a histone, and it calls around various histone, and you open it up and you see that that is one DNA, right? So that is chromatin, is DNA. Now, when the cell is preparing to divide or is dividing, then you see one DNA as chromosome. And this is what happens. So this is the chromosome. This is what happens. You have this one DNA, and this DNA duplicates. You have one DNA opens up, and the whole DNA will duplicate. So it does that gradually. It opens up. We'll discuss this process too. It opens up, and you see this adenine, you have its complementary base. Thymine comes to join. You have another thymine here and adenine. So it opens up, and a complementary base pairing, another DNA is formed by its side, and this one too. So the original DNA, you end up by having two. This is the original. The original is the blue colored DNA, that is it, and the blue colored DNA, and it duplicated, replication it's called, and that becomes the red. And these two are held together at a point called centromere. And at this, you can see it neatly as chromosome. And so you see it as chromosome, and this chromosome is just one DNA that has replicated, and we call these two as sister chromatids, right? And its centromere is holding it here, and that's a chromosome. So that is what happens. So if we look at this chromosome here, and this chromosome is unraveled, you see that the chromosome is thread-like, and it goes and it's open up and open up. Now you can see the histones that forms this DNA, and is the same with this one is also unraveled and open up. It will be found out that it is the same DNA, sister chromatid. So this is one DNA. All right. So that is what happens. You see DNA as chromatin material or chromosome is one and the same DNA. Good. Now, we want to find out what makes DNA universal, same in all organisms. And yet DNA is also different in different organisms. Look at it. DNA is universal in the sense that it's the same for different nucleotides that form DNA in all organisms. The same 4 nucleotides with adenine, guanine, thymine, cytosine, same 4 nucleotides. And it's the same nucleotide pairing. Adenine always pairs with thymine, guanine always pairs with cytosine in all organisms. And in all organisms, the genetic code, that is the sequence of bases that stand for one amino acid, is the same. So the sequence of bases, you have thymine here, you have cytosine here, you have adenine here. These 3, if they code for one amino acid, is the same code that codes for an amino acid in all organisms. So it's the same 4 nucleotides, the same nucleotide pairing, the same code, genetic code that coding for the same amino acids in all organisms, and it's the same 20 types of amino acids in all organisms. But yet, organisms are different. So what is it that makes organisms different, even though the DNA is basically the same? Well, one is that the numbers of chromosomes or the numbers of DNA is different in every organism. Different organisms have different numbers of chromosomes. So every different organism, that is a species, has its own numbers of DNA or chromosomes. That is the chromosome number. It's an identification number for every organism. So for instance, human beings have 46 chromosomes and it's 23 pairs. We'll explain. The dog has 78 chromosomes and is 39 pairs. The cat here has 38 DNAs or chromosomes and is 19 pairs. Well, this fruit fly, Drosophila, has 8 chromosomes, 4 pairs. Then we have this toad that has 26 DNAs, 13 pairs. So different numbers of DNA in different species. Another thing about DNA that is different in different organisms is that the arrangement in the sequence of bases in the DNA are different. For instance, we have these DNA pieces. You see, the way the bases are arranged, the sequence, you have thymine here, cytosine here, adenine here. You can have different sequence as soon as even one change. And instead of you have thymine, adenine is changed. It will be different. And it will produce a different amino acid and finally ends in a different protein. And so it will be a different variety. And finally, many such differences in sequences of bases will lead to a different organism. Not only that, the shape of the DNA, see, here we have a double helix structure of DNA. Usual. The shape, the size of the DNA can be short DNA, it can be long. The shape, the sizes of the DNA is different in the different organisms. And not only the shape and the sizes, the way the DNA is turned around. For instance, look at this bacterium. This bacterium has a circular DNA and it has some plasmids here, which also looks circular. So these are the reasons that makes DNA different in different organisms. Well, we have other things to talk about. Which cells of the organism has DNA? You have organisms like a human being. Researchers have found out that human beings have about 30 to 40 trillions of cells. And every living cell has DNA in its nucleus. If a cell has no DNA in its nucleus, it means the cell, when it started as a cell and it was living, had DNA. But with time, it lost its DNA. It may be dead or it just lost it. All right, but every living cell has DNA. So this human being with 30 to 40 trillions of cells, every cell of it has DNA. So we come to look at human beings, which is the eukaryotes, and we talk about eukaryotes and prokaryotes in our lesson on bacteria, and check on our lessons also on viruses. We explain what eukaryotes and prokaryotes are in those lessons. So every eukaryotic organism has two major types of cells. One type of cell is the body or somatic cell, and the other type is the reproductive cells. So I have this young man here. It has these two types. This blue are body cells, and this young lady here, the blue cells are body cells. And then reproductive cells. Reproductive cells, the sperm is a reproductive cell, sex cell. It also has DNA. And the egg is also a reproductive cell, which have DNA. Now, if the organism is diploid, like a human being, then it has DNA, maternal and paternal. It has two. So each body cell, or the blue cells, body cells, have DNA, but two sets of DNA or two sets of chromosomes. And the reproductive cells, which is the sex cell, egg, or sperm, has only one set of DNA. So this human being here, the body cells, all the trillions of body cells, each body cell has 46 chromosomes or 46 DNAs in its nucleus. And the 46, they stay as 23 pairs in every body cell, every one of them. And the sex cells, that are reproductive cells, the sperm, has 23 DNA or chromosomes. And the egg too has 23 DNA or chromosomes. So cells of organism, every cell has DNA. Well, this is what we end on today. The lessons have been fruitful. Please give us a thumb and subscribe. In our next lesson, we will still have a short overview of DNA before we launch in to look at the detail of all the areas we have talked about. Thank you very much. Goodbye.